Hi everyone, my name is Maria Camila Medina, and first of all, I just really want to start saying especially thank you to the EvoDevo Symposium organization on the Congresso Nacional de Botânica do Brasil for giving me the opportunity to present these results. I'm very excited to be here and I hope that you can enjoy this talk. You can find me in the Twitter, and this is my email if you want to contact me at any time. This work was developed in the Bioscience Institute at the University of São Paulo by myself, the Dr. Mariane Sousa-Baena, Professor Mariane S, and Professor Diego Demarco. Okay, let's go. Well, I would like to start this talk saying that lettuce bearing plants are very diverse and we can find them as big trees, shrubs, lianas, herbs, and they are distributed in the more variable habitats and belonging to diverse taxonomic groups, as we can see in this vascular plants tree. We have laticefers plants that are ferns, gymnosperms, and they are widely present in angiosperms, and you can see marked with the blue arrow. And they are in groups that some of them have no close phylogenetic relationships, and so we can observe a case of convergent evolution. In the tropic, we find a greater diversification of latex-bearing plants, and that coincides with the high diversification of insects in the same regions. It is widely known that laticifers are defense secretory structures that produce and accumulate primary and secondary metabolism products which form an emulsion whose composition can vary depending on the taxon. But it is not only the latex that presents this variation in its composition, because structurally, laticifers can present differences too. Well, Fan defined laticifers as cells that produce latex, and Demarco classified laticifers into two types, non-articulated and articulated. The non-articulated ones consist in a single cell that will grow indeterminately in the plant body, and that in some species can be branched. The articulated ones are a row of cells in which the terminal walls of each cell can remain entire, or in some groups the terminal wall can be degraded. That degradation can be partial or complete without leaving traces of wall. And additionally, in some groups, lateral degradation can occur producing branched laticifers. With respect to origin, in the primary growth, laticifers are originated in ground meristem and or procambium, or both, depending on the species. And in the secondary growth, laticifers are originated from the activity of the vascular cambium. With respect to the type of growth of laticifers, many authors have been described that laticifers can grow into two ways, intrusively or by cell addition with posterior cell fusions and anastomosing species. The apical intrusive growth has been related with the non-articulated ones and the cellular addition with the articulated ones, and I will explain each one. According to Malberg, the intrusive apical growth is characterized by occur in cells with unicellular origin. I am showing to you an example in an embryo because laticifers start their development from the embryo, and in fact, laticifers are present in all organs of the plant except in ovules and seed coat. In the nodal plexus, laticifers will be originating in the single cell will have an intrusive bidirectional growth. because laticifers would degrade the middle lamella from neighbor cells by secreting digestive enzymes, and in this type of growth will only occur nuclear divisions. Laticifers also will grow in intercellular spaces. In the case of cellular addition type of growth, laticifers have a multicellular origin as the almost all type of plant cells. Here I'm showing to you this example with a shoot-apical meristem which is growing in the growth direction. New laticifers are differentiated from ground meristem or procambium depending on the species, and new laticifers differentiated are added to the latiferous system. In anastomosing laticifers, the terminal wall degradation also occurs. With respect to classification, I want to show you the case of Apocynaceae, when laticifers were classified as non-articulated, but more recently, authors showed that the region of the plant where laticifers were analyzed was already a developed region, a mature region. And actually, these laticifers are articulated anastomosing that also are branched. In this case, this is the type of laticifers that lost their terminal walls entirely. And this not only occurs with Apocynaceae, but with another families too, that have also anastomosing laticifers that are branched. And currently we are doing a work analyzing families described as having non-articulated laticifers, because our hypothesis is that non-articulated laticifers do not exist. And for this, in this work, we focused in articulated laticifers. Even though some studies to reclassification of laticifers have been made, the type of growth is an aspect that still generates some confusions. Here I'm showing to you an example, also in Apocynaceae, when some author describes articulated anastomosing laticifers as having initially normal cell divisions, but after laticifers lost their terminal walls, they will be growing intrusively in the tips. The combination of two types of growth in the same type of plant cells was already described for phloem and xylem fibers. But other authors say that the tips are actually an effect due to thinking in 3D dimensions. Laticifers do not form straight rows, but they have a very sinuous growth. And this is a problem of analyzing tissues that grow in a 3D plane from a 2D plane. And this is made with classic anatomical techniques that have helped a lot to understand the developmental process of laticifers, but have this kind of limitation. In order to understand the type of growth described in general for plant cells, we observed that from the perspective of plant anatomists, plant cells grow in three ways, and it depends on the plant cell type. According to Fend in SAU, plant cells have three types of growth: the symplastic growth, the protrusive growth, and the intrusive growth. On the other hand, it is known that cytoskeleton plays an important role in the determination of the type of growth in plant cells, and plant scientists that study plant cell growth, analyzing the cytoskeleton organization, establish that plant cells can grow in a diffuse or polarized way. And this type of growth corresponds with the type already mentioned by plant anatomists based in structural observations. So, with respect to diffuse type of growth, this occurs in two ways. isotropic and anisotropic. The isotropic type is observed in meristematic cells in root zone of elongation, which are cells that are in differentiation. The anisotropic type is common in hypocotyl elongation and pavement cells, which are cells that are in elongation process. In all of these, the cortical cytoskeleton, here marked in green, have a transversal pattern with respect to growth axis. This is consistent with synplastic growth proposed by Fan and Esau that occurs in that type of cells and regions of the plant. With respect to protosieve growth, this is a type of polarized growth that occurs in trichomes and root hairs where cortical cytoskeleton are arranged parallel to axis growth. And finally, the intrusive growth that is considered an extreme form of cell growth occurs in pollen tubes, but also have been described for internal cells are sclerites, phloem, and xylem fibers, and laticifers as a question because this is where we want to respond. The cortical microtubules have also a parallel arrangement with respect to the cell growth. Well, so taking all into account, we return to our initial question. So, how do laticifers grow? And for that, we did two experiments in shoot apices of adult plants. First, we performed a developmental analysis using usual techniques in plant anatomy. And also, we performed a microtubules immunolocalization experiment using anti-alpha tubulin antibody and confocal laser microscopy to image and analysis. Then we selected three types of articulated laticifers, two species with laticifers that are not branched. The first one was Urviliol masse of the Sapindaceae family with articulated non-anastomosing laticifers, and the second one was Hypomianil of the Convolvulaceae family with articulated anastomosing laticifers. Both species were never described as having laticifers with intrusive growth. Also, we selected Asclepias curaçavica for Apocynaceae family, a family with laticifers described as having intrusive growth. As results, we observed that in species with articulated laticifers that not branched, they are originated in ground meristem. And as described for symplastic type of growth, the cell walls were always observed attached to each other during all developmental process. In the non-anastomosing, the terminal walls were always entire, and in the anastomosing, the degradation of terminal walls occurs latterly, and we don't observe intercellular spaces in this species. With respect to cortical microtubules arrangement, we observed that in both types of laticifers, cortical microtubules are arranged perpendicularly to the cell growth axis. And the same pattern was observed in idioblasts present in Urviliol masse and parenchyma cells in both species in development areas. We do not have records from parenchyma cells and idioblasts as having intrusive growth. In the case of Asclepias curaçavica, which have articulated anastomosing laticifers that also are branched, laticifers are originated in ground meristem in procambium, and the differentiation process occurs very fast And just below the primary stem, we observed laticifers with thickened cell walls and latex already being produced. Here we can see the laticifers differentiating from Procambium with terminal wall still present, and we can see also the formation of a ramification. And in the same area, but in the ground meristem region, we observed a laticifers that already lost their terminal walls and with a great quantity of latex. It is very important to highlight here that in this species we also don't observe intercellular spaces, and the cell walls were always observed attached to each other during all developmental process. With respect to cortical microtubules arrangement, we observed that Asclepias corazavica have laticifers with cortical microtubules arranged perpendicular to the cell growth axis. The same pattern was observed in parenchyma cells in developing areas, remembering that we don't have recourse for parenchyma cells and as having intracellulary growth. In this summary image, you can see the three types of laticifers, and independently if they are branched or not, or if their terminal walls are degraded early or late, or just are not degraded, the pattern of cortical microtubules was the same in the three species, as you can observe here. And for that, we have the conclusion that the cortical microtubules are organized in the same way in the three type of laticifers, presenting a pattern perpendicular to the cell growth axis, which is typical of cells that grow diffusely. It suggests that laticifers grow diffusely. All this work was recently published in the Frontiers in Plant Science, in the Plant Development and EvoDevo. And if you want to know more about this work and the details, I would like to suggest to read the paper. And well, finally, I would like to remind you that laticifers arose several times in the history of vascular plants, and this is evidenced by the morphological and structural differences that we find in the different groups. But the way in which plant cells grow is a highly conserved characteristic in vascular plants in the different cell types. Well, we have the case of intrusive growth in pollen tubes, in the searching of ovules, and the combination of the two types of growth in the sclerites. But actually, we need to do more studies in other groups. But so far, we see that the type of growth in laticifers is shared by laticifers present in distant lineages, as you can see again in this tree, and with different structural characteristics. But with respect to the type of growth, it seems that is a shared characteristic. And well, thank you again so much for your time and for listening to me. And I hope you have been curious about laticifers. And I would like to say thank you again to Dr. Diego Demarco from Laboratory of Plant Anatomy in the University of São Paulo, to Dr. Mariane S. From Genomic and Transposal Elements Lab in the University of São Paulo too, and to Dr. Mariane Sousa-Baena, who is a postdoctoral researcher in Cornell University. Thank you so much, and I'm open to questions.